Hi everyone, my name is Foad Hamidi and I'm going to present our paper entitled Turning the Invisible Visible, Transdisciplinary Bioart Explorations in Human DNA Interaction on behalf of my colleagues Lydia Stamato, Lisa Scheifele, Rian Hammond, and Nisa Asgarali-Hoffman. Bioart is an art practice in which artists work with living organisms, including animal, plants, fungi, and others, for artistic and creative purposes. Some examples include Alba, in which the artist genetically modified a rabbit to glow in the dark, and Nature, in which the artist worked with the development process of butterflies to create patterns on their wings, and a contest run by the American Society of Microbiology in which contestants can draw on agar with colorful microorganisms. The contribution of this paper is twofold. First, we investigate the current perspectives of bioartists on working at the intersection of science and art. And also, second, we report on a first-hand account of how to creatively interact with living organisms on a molecular level through current do-it-yourself synthetic biology methods. These contributions add to the conversation at the intersection of human-computer interaction and bioart. For the first part of our paper, we interviewed 6 bioartists. We found that bioartists often describe their work in terms that invoke skills and epistemologies that cross disciplines towards a shared collaborative goal, a quality that we identify as transdisciplinary fluency. For example, P1 said, "I wanted to develop an art piece that was in effect a biosensor, a meter of whether the genes had been synthesized. We're trying to do it in E. coli, and we're working with designing the genetic constructs to create the different steps of that pathway." Another finding was that bioartists pointed to a sense of familiarity, including through cultural symbolism and the shared quality of being alive or supporting life with the organisms that they work with. This quality contrasts with the DNA as code metaphor that sometimes is used in synthetic biology. For example, P5 said, "As I told you, you're working with another living organism. You're also a living organism." Finally, we found that bioart often requires a specialized knowledge and equipment, necessitating artists to work together with scientists to negotiate access, and that this negotiation in turn increases access to biology for diverse audiences. In the second part of the paper, we describe our experience with human DNA interaction through creating transgenic poetry-infused wine. The project revolves around the famous line of Sufi poetry by Hafiz, the 14th century Persian poet, that revolves around themes of transformation and persistence through love. هرگز نمیرد آن که دلش زنده شد به عشق، ثبت است بر جریده عالم دوام ما. In this project, we first encoded the poem into Morse code and then into a viable DNA code. We then fabricated this DNA code and inserted it into a plasmid backbone. After amplifying the DNA and verifying that the resulting plasmid included the sequence with the poetry using an enzyme digest on an electrophoresis gel, we transformed the linearized DNA with living yeast cells and selected for yeast cells that contained the poetry sequence. We then grew the yeast overnight and combined it with grape juice to make transgenic wine. Raz pays homage to the resilient spirit of Persian mystic poets who used the power of metaphors and linguistic transmutations to capture vivid experiences of personal and spiritual transformation. Additionally, its engagement with the process of winemaking, which is taboo in this cultural context, parallels the ethically ambiguous practice of genetically modifying living organisms. In conclusion, we found that bioart offers exciting modes of inquiry for human-computer interaction that are inherently transdisciplinary and can be characterized as art research. Additionally, we found that interacting with living organisms at a molecular level can provide insight on structures of access and power that surround our relationship with them and the environments we live in. Thank you very much. I'm happy to answer questions via email, and please see the paper for more details.